Every living organism, from the smallest bacterium to the largest whale, carries within its cells an extraordinary instruction manual. This manual, known as deoxyribonucleic acid, or DNA, is the fundamental blueprint of life. It dictates everything from our eye color to our susceptibility to certain diseases. For centuries, the intricate mechanisms of heredity remained a profound mystery. The discovery of DNA structure in the mid-20th century marked a pivotal moment in scientific history. It opened the door to understanding how life's information is stored and transmitted. Unraveling the secrets held within this molecule has transformed our understanding of biology. It has paved the way for groundbreaking advancements in medicine, agriculture, and forensic science. The journey to decode DNA is a testament to human curiosity and ingenuity. At its core, DNA is a remarkably elegant molecule. It forms a double helix, often described as a twisted ladder. Each rung of this ladder is composed of two chemical bases, always pairing in a specific way. Adenine, or A, always pairs with thymine, or T. Cytosine, or C, always pairs with guanine, or G. This precise pairing rule is fundamental to how DNA stores and replicates information. The sequence of these base pairs along the DNA strand is the genetic code. This code contains all the instructions necessary for an organism to develop, survive, and reproduce. It is a universal language shared by nearly all life forms on Earth, highlighting our common evolutionary heritage. How does this seemingly simple sequence of A, T, C, and G translate into the complexity of a living organism? This process is explained by the central dogma of molecular biology. It describes the flow of genetic information within a biological system. First, DNA is transcribed into messenger RNA, or mRNA. This single-stranded molecule carries the genetic message from the DNA in the nucleus to the ribosomes in the cytoplasm. Next, the mRNA is translated into proteins. Ribosomes read the mRNA sequence in 3 base increments called codons. Each codon specifies a particular amino acid, the building blocks of proteins. Proteins are the workhorses of the cell, performing a vast array of functions. They form structures, catalyze reactions, transport molecules, and transmit signals. Thus, the DNA sequence ultimately dictates the proteins an organism produces, shaping its traits and functions. The ability to read or sequence DNA has revolutionized biology. Early methods like Sanger sequencing, developed in the 1970s, were groundbreaking. They allowed scientists to determine the order of bases in relatively short DNA fragments. The Human Genome Project, launched in 1990, was an ambitious undertaking to sequence the entire human genome. Completed in 2003, this monumental effort provided the first comprehensive map of human genetic information. It laid the foundation for modern genomics. Today, next-generation sequencing, or NGS technologies, have dramatically accelerated the process. These high-throughput methods can sequence millions of DNA fragments simultaneously. This has made DNA sequencing faster, cheaper, and more accessible than ever before. NGS platforms generate vast amounts of data, allowing for the rapid analysis of entire genomes. This technological leap has opened new frontiers in research, diagnostics, and personalized medicine. Decoding DNA has profound implications across numerous fields. In medicine, it enables the diagnosis of genetic diseases, sometimes even before birth. It also facilitates personalized medicine, tailoring treatments based on an individual's unique genetic makeup. Pharmacogenomics, a branch of personalized medicine, uses genetic information to predict how a patient will respond to specific drugs. This minimizes adverse reactions and optimizes therapeutic outcomes. In forensics, DNA profiling is an indispensable tool for identifying individuals and solving crimes. Its unparalleled accuracy has revolutionized criminal investigations and exonerated the wrongly accused. Agriculture benefits from DNA sequencing through improved crop yields and disease resistance. Genetic insights help develop more resilient and nutritious food sources, addressing global food security challenges. Furthermore, decoding DNA allows us to trace evolutionary relationships between species. It helps us understand biodiversity and the history of life on Earth. Despite incredible progress, the journey of decoding DNA is far from over. Ethical considerations surrounding genetic privacy, discrimination, and the responsible use of genetic technologies remain paramount. The sheer volume of genomic data generated presents significant computational and analytical challenges. Developing robust bioinformatics tools and data storage solutions is crucial for harnessing this information effectively. Looking ahead, technologies like CRISPR gene editing offer unprecedented precision in modifying DNA sequences. This holds immense promise for correcting genetic defects and treating diseases. Synthetic biology aims to design and build new biological parts, devices, and systems. This could lead to novel applications in medicine, energy, and materials science, pushing the boundaries of what is possible. The ongoing quest to fully understand and utilize the blueprint of life continues to inspire scientists worldwide. It promises a future where our knowledge of DNA will unlock even greater potential for human health and well-being.